Imagine spending years developing a generic peptide drug, clearing every toxicology hurdle, optimizing your synthesis, scaling your process, and then your regulatory dossier gets kicked back. Not because your molecule is wrong, but because you couldn't prove it was the same. This is the reality of many generic peptide developers today, and it's entirely avoidable. Hi, I am Raghuveer Singh Parmar, Director Scientific Affairs at Resolve Mass Laboratories Canada. Welcome at Resolve Mass Laboratories. We bring deep scientific expertise in advanced analytical characterization and complex molecule development to accelerate your IND and ANDA programs. Let's start with the why. Regulatory agencies like the FDA don't just want to know that your molecule looks like the reference product. They want unequivocal proof that it is the structurally equivalent at every level. And this is where many teams underestimate the complexity. Molecular weight alone, not enough. Think about it, a single amino acid substitution, a stereochemical flip from L to D configuration, or an unresolved cyclic variant can fundamentally change how your peptide behaves in the body. The safety profile shift, the pharmacology also shift, and now you have a different drug even if the sequence appears correct on paper. So sameness studies aren't just a checkbox, they are a regulatory prerequisite, and they demand a very specific kind of evidence. This is where orthogonal methodology comes in. The gold standard approach integrates three complementary techniques, and I want to emphasize the word complementary because it's the key. LC-MS tells you about primary structure, the amino acid order. The peptide mapping validates the fragment profile and catches impurities, and NMR goes deeper. It confirms stereochemistry and three-dimensional conformation. Together, these three techniques cover dimensions that no single method can cover alone. Here is the question worth sitting with. Why can't mass spectrometry do it all? Mass spec is really powerful. It's the backbone of sequence confirmation, but it has a fundamental limitation. It doesn't see space. It can't reliably distinguish D amino acids from L amino acids. It can miss certain cyclization patterns. It can't resolve some disulfide bond arrangements. Chromatography adds another layer, separating degradation products and process-related impurities that might collude otherwise. And NMR spectroscopy resolves atomic arrangement and conformation at a level nothing else can match. When you combine all three, you are not just generating data, you are building a wall of evidence that answers a reviewer's questions before they can even ask them. And in our experience at Resolve Mass, that kind of proactive, comprehensive characterization is precisely what accelerates review timelines. Let's get into the mechanics. In LC-MS sequencing, the intact peptide is first separated by liquid chromatography, then subjected to high-resolution mass analysis. The real magic happens at the fragmentation step. MS breaks the peptide apart, generating what we call B and Y ions, and we reconstruct the amino acid sequence from those fragments like reading a molecular puzzle. What we are confirming here, the correct sequence, any substitutions or truncations, post-translation modification variations like oxidation or deamidation, and overall mass consistency against the reference product. This is the cornerstone of primary structure confirmation in any sameness dossier. Peptide mapping takes this a step further. You enzymatically digest the molecule, separate the resulting fragments by LC, detect them by MS, and then this is the elegant part. You overlay the chromatography of your test article directly against the reference product. What you are looking for is reproducibility of that fragment profile. Any deviation, any extra peak, any missing fragment, those are the signals worth investigating. Peptide mapping is how you catch micro heterogeneity, subtle sequence variants that might slip past intact mass analysis. In complex molecules, lipopeptides, cyclic structures, this kind of fragment level scrutiny is non-negotiable. And then there is NMR, the technique that takes us to the atomic level. This is where I see many teams either underinvest or get nervous, because NMR can feel like a black box if you haven't worked with it extensively for peptides. But here is what NMR uniquely delivers: stereochemical conformation, D versus L amino acid configuration, cyclization integrity, whether ring formation occurred correctly, disulfide bond validation confirming not just presence but pairing and positioning, and conformational isomer differentiation, which is critical for peptides with flexible backbones. In challenging molecules like semaglutide analogs, cyclic peptides, lipid conjugated structures, we have seen cases where MS looked perfectly clean, but NMR revealed a stereochemical impurity that would have been invisible otherwise. That's the kind of gap NMR closes. Regulators have become increasingly sophisticated in what they come from sameness packages. Comprehensive orthogonal data reduces the back and forth, fewer review questions, faster progression, a stronger submission overall. Think of it this way: every piece of orthogonal evidence you include is one less deficiency later you receive. That's not just a scientific argument, it's a business case. Of course, the path is rarely straightforward. In practice, the most common challenges we encounter are detecting low-level sequence variants that require high-resolution MS to even see, characterizing labile modifications like oxidation and deamidation that degrade during sample preparation if your methods aren't optimized, working with cyclization patterns and lipid chains that demand specialized digestion workflows, and achieving definitive stereochemical purity assessment, which, as we have established, really does require NMR. The strategic response to each of these challenges is method optimization, not just method selection. High resolution LC-MS, MS, tailored digestion conditions, and well-designed NMR acquisition strategy collectively close the gaps. So this brings to all together, demonstrating sameness for a generic peptide is not a single technique problem. It never was. LC-MS gives you the sequence confidence. Peptide mapping gives you the fragment level integrity and impurity resolution. NMR gives you stereochemical and conformational certainty, and together they give you regulatory confidence. And if you'd like to talk through your specific molecule and what a robust sameness package would look like, reach out to us. These are exactly the conversation we have every day. Partner with Resolve Mass Laboratories to overcome your toughest drug development challenges and move your program forward with scientifically robust solutions. Thank you so much for watching this session, and goodbye for now. See you in the next one.